Right. So we are picking up with chapter 12, and right when we left off of class, we were starting to get into exactly how an impulse is transmitted. So, dealing with impulses within nerve cells, you kind of have to think about where everything is positioned. So, you have your nerve cell, and just like any cell, it's going to have a cell membrane that's going to enclose it and keep the internal components on the inside and the external components on the outside. And it's really that separation between the inside and the outside, and that barrier of the cell membrane that is going to cause for an impulse to actually be able to take place when the need arises. By having the separation of the cell membrane, you have the ability to then control when your nerves fire, when they send impulses to other nerves or to muscles or to other body parts. And so the cell membrane of a nerve is very important because it regulates you and makes sure that you're not constantly firing off impulses or reacting to every little thing that comes in contact with your body. As we talk about the nerve impulse, I introduced some of the terms earlier today. So we talked about polarization, we talked about depolarization, and then repolarized. And as we move through the packet, there are going to be some additional terms that we need to introduce. And the top of page 8 actually starts off with one of those terms. And it talks about transmembrane potential. And if you break this word down, potential, ability to do work, membrane, we already have identified that we're talking about things that are separated on either side of the cell membrane. And then trans just simply tells you that you are moving across or you are spanning through something. So when we talk about transmembrane potential, it is the potential difference measured across a plasma membrane and expressed in millivolts. So I wanted to introduce the concept and give you guys the visual of the positive and negative charges on the inside and the outside of the nerve membrane. And because you have those different charges, you do have an electrical charge within your nerve cells, and that is measured in millivolts. In normal conditions, your internal part of the nerve cell, remember, you have the positive ions on both sides of the cell membrane, but on the inside of the cell membrane you also have these very large sulfate and phosphate ions. They outnumber the positives, and so the inside of the nerve cell will give off a negative charge. And at resting potential, which is just a fancy way of saying that the nerve cell is not conducting an impulse, so resting potential, your nerve cell is going to be at about 70 millivolts. As we talk about nerve impulses, we're going to be dealing with ions, and I also kind of reintroduced those. You guys seem to have a very good handle on what ions are. But ions are any particle that carry a charge with them. And within the nerve cell, you are going to have these channels or these passageways through the cell membrane, because you have to get these ions to be able to interact with each other at the appropriate times so that you can cause a nerve impulse to actually take place. So when you don't want an impulse to fire, you are going to keep those channels or those gates closed, and when you do need it to fire, then you're going to open up those gates and allow the ions to interact with each other. Couple different types of channels that you have. The first one it introduces is the passive channels, and these are referred to as leak channels. Passive channels, and if you go back to when we talked about passive movements across cell membranes, passive just simply meant that the flow of ions is the direction that you want it to go, the natural flow of movement, and so it's not going to require any energy to take place. These passive channels are also known as leak channels because they're always open, and so there's going to be the movement of molecules constantly through that particular channel. And it mentions that permeability will change with conditions. And so as conditions in and around the cell become altered, how many ions are flowing or leaking through that channel will vary. The second type of channel that you have are active channels, and these are gated channels. And active indicates that there is work that needs to be done in order to get these to work the way that they need to. So gated channels means that you're going to have some type of regulation, that it's not just passage at will through the gates or through the channels. There has to be something that is going to cause the gates to open up and allow for the movement of the molecules from one side to the other. There are three different categories of gated channels. You have the chemically gated, the voltage gated, and the mechanically gated. And the next three slides just kind of give you a brief explanation of those different type of channels. With the chemically gated channel, it is going to only open up if you have the right chemical in the presence of that particular gate. And so you could have a variety of substances passing through that channel, but until it's the right chemical, that particular gate is going to remain closed. So kind of think of it as a lock and key system. You can try a lot of different keys into a lock, but until you have the right one, it's not going to open. The second type that you have is the voltage gated channels, and this is going to be what we deal with quite a bit when we talk about nerve impulses. And simply what's going to happen here is that there's going to have to be a change in the charge across the cell membrane in order for those gates to open up and allow for the movement of the ions. And we'll talk more about that as we get further into the chapter. And then the last one is the mechanically gated channels, and it uses the term distortion, that the membrane is going to respond to some type of distortion. And essentially what they're talking about is if you just have common touch, pressure applied to the skin, some sort of vibration, that is what they're talking about when they mean distortion. So it's a little bit different term that what we would typically use or hear to refer to things like touch, pressure, and things of that nature. Flip over to page 9. Page 9 starts out with resting membrane potential. And I already mentioned that when everything is in its starting positions in the nerve cell, so no impulse, everything is just positioned at its starting points, the inside of the nerve cell is going to register at about negative 70 millivolts. And this is because of those negatively charged particles that you have on the inside of the nerve cell. The extracellular fluid, abbreviated as ECF, and the intracellular fluid of a nerve cell are going to vary very greatly in the ions that they have present. And up until this point, I've just been talking about positive and negative charges because I wanted you to get that concept of going back to the laws of attraction and thinking about how positive and negatives are going to relate to each other. You start introducing letters and things like that, and the focus kind of goes other places. So those positive charges that I was talking about on the outside of the nerve cell, those are actually sodium ions. And the positives that you have on the inside of the nerve cell are potassium ions. So resting potential, the sodiums are going to be on the outside of the nerve cell, potassiums are going to be on the inside of the nerve cell. As things start to take place and a nerve impulse starts to register within that nerve, what you're going to see happen is you're going to start exchanging those sodiums and those potassiums. So they're going to start moving from their original positions to flip-flop. So the sodiums, which start on the outside, and the potassiums, which start on the inside, are now going to exchange places with each other. But what happens is you have three sodiums excuse me, that are going to be transferred